No. Boy, have I got fun for you guys. It's actually partially problem session tomorrow, so we're going to go over some things. Yeah, I'll let you that. See if I can do this without. I got it over there. I'll put the rest of them here. People can grab one as they come in. And now I need to find my. You guys want to learn about the plasma membrane? Probably not. Why is it showing me and not the PowerPoint? Okay, fix that. There we go. That's better. All right. So I started working on your exams. I should say I started thinking about working on your exams, and I did do some preliminary work, but I realized I was so bloody tired that I would just decide to go home. So I will work on your exams tonight. I was hoping to have them done by now, but so let's start with a little bit of history here. So early on, there was we had evidence from X-linked genes and chromosomes that genes resided on chromosomes, but we didn't have any real definitive evidence. It was pretty good, but what people wanted to see was they wanted to physically see chromosomes that they could tell apart and assign genes to. And two people really were involved in that. The first one is Barbara McClintock, and she worked with corn. If you guys want to pick up a guy up here. So Barbara McClintock counted corn kernels in millions of them, I think. But she and working with corn found this, and another biologist working with our favorite molecular model, Drosophila, Kurt Stern, was able to do this. And we're going to look at the Kurt Stern's experiment here. I've got it drawn in my notes, but you can probably see it better up here if I turn the lights off on the. Okay. So why is this one dim and that one bright? I'm gonna go to the bright one. This one needs caffeine, that one doesn't. Okay. What he did is he found these X chromosomes. So he's looking at, I'm sorry, these aren't X chromosomes. These were, the word will come to me in about 5 million years. These are autosomes, not sex chromosomes. Okay. He found these sex chromosomes that had these physical defects that he could identify the chromosome and from its homologue. What we wanted to be able to do is track the homologs and see if the traits move with the homologs. So he had this one, and it had this discontinuity in it. And it carried the carnation eyes mutation, which is a recessive mutation, and it carried the bar mutation, which is a dominant mutation. Then he had another chromosome where a piece of the Y chromosome had broken off and been stuck to that chromosome. That happens sometimes during repair. So he has this homolog that has the wild type carnation and the wild type bar. In other words, this is all wild type, right? And it's going to undergo meiosis. And without crossing over, he gets the parental genotypes or phenotypes. So here we have a fly that's wild type for carnation eyes, but that has bar eyes because bar is dominant. And Drosophila, capital letters mean dominant alleles. And then over here, this fly is also got red eyes and bar eyes. And that's exactly what we started with, red eyes and bar eyes. So those match the parents. Oh, great, my hearing aid's dying and spitting noises in my ear. You don't care about that. But you also get crossing over, and we get these recombinants that we can see that you switch the genes. And he can actually physically see the recombination results because of the discontinuity and the bit of Y chromosome on the homolog. So this was really the evidence that was really, okay, yeah, genes reside on chromosomes. All right. So let's talk a little bit more about this idea of physically what happens during crossing over. Physically, at the beginning of meiosis prophase, the homologs find each other and the synaptonemal complex forms, and we form the tetrad. So the synaptonemal complex keeps them nicely together, and then they can start their crossover. But once the synaptonemal complex dissolves, which is in prophase 1, it comes together and it goes away by the end of prophase 1, they're still held together because the sisters are each held together with cohesin molecules. So these are proteins that hold the sisters together. And since we have swapped parts of the chromosomes, they form this conformation that kind of looks like an X. Here are our centromeres there and there. So this object can be pulled apart. Once the cohesin is broken, then this is going to be one chromosome, this is going to be the other chromosome. So we have these guys together. That's basically what's physically happening in crossing over. Now, we're going to, in cell biology, go into strand invasion and all sorts of stuff because it's not a random process insofar as how it's done. However, it can happen anywhere along the chromosome. It's a very well-regulated process. In fact, there is a mechanism that I think I have it down here that we're going to call interference, and we're going to calculate interference, excuse me. And interference ensures that a chromosome has at least one crossover before it lets meiosis go on. And that happens in almost all sexually reproducing organisms. All right, anything else I want to say? Tetrad, synaptonemal complex. So yeah, they kind of show it here. This is, these are tetrads right here. Hard to see, but that's prophase 1 diplotin. The synaptonemal complexes come together and brought the homologs together. All right. So let's start some math. That's a big jump down. I went from there to recombination frequency. Let's see what I have here. All right. So we talked briefly about how we calculated map units or centimorgans. One map unit is equal to 1% recombination frequency. And one map unit is equal to 1 centimorgan. They're all synonymous. All right, and we looked at W and Y, white eyes and yellow body, and white eyes and miniature wings. And these are all on the X chromosome. And we found that there were 1.1 map units between white and yellow, and that there were 32.8 map units between white and miniature wings. So we can actually use this kind of information to figure out the relative position of genes. And so I'm going to come off to my notes here for a second and talk about some of the things that I have in my notes. First of all, let's define recombination frequency. And we're going to say that's equal to recombinant offspring over the total number of offspring times 100. Now, the times 100, we're actually making a percentage, but as you recall, 1% RF equals 1 map unit equals 1 centimorgan. So we can just multiply this by 100 and call it map units or centimorgans. We don't have to put the percent in there, but this actually is a percentage. We're just converting the decimal to a percentage. And then I've got some acronyms I'm going to use. The first one is NCO. Non-commissioned officer. No, that's not it. That's the military. It's no crossover. The next one is SCO. Single crossover. And then we have DCO. So we're going to be looking at recombination of genetic, I don't know where I put the spelling. Synthetic, I'm probably misspelling that and mispronouncing it right now. My brain is fried. I'll find it later. Genes, basically genes on the same chromosome. We have all sorts of synonyms that we use all through genetics. It's great. All right, so we have no crossover, single crossover, and double crossover. Can triple crossovers happen? Probably, but double crossovers are rare. So I would think a triple crossover, if you were to detect one, would be uber rare. These are rare events. Okay. So what I want to talk about, the recombination frequency of genes on separate chromosomes is 50%. In other words, we're going to get half parental and half recombinant from a straight dihybrid cross. They're not on the same gene, same chromosome. We're talking about genes. I should be using the term loci. Anyhow, the genes or loci are not on the same chromosome. So we're going to get 50% parental, 50% recombinant. And so let's look at that just to make sure that sticks in people's brains. All right, so let's say we have our F1 and it's A, B, B, A, B, B. So we have a homozygous dominant and a homozygous recessive that we're crossing for our F1, or our parent. That's not our F1, that's our parent. Our F1 are 100% going to be heterozygotic for both loci, right? You know that. That's just a straight dihybrid cross. Now, if we look at the gametes that these flies can produce, whether they're males or females, the gametes they can produce would be dominant, which is a parental phenotype. What I just do it the way I have it here. I put the other parental next. They can be recessive or homozygous recessive, like we have here. But they can be recombinant. We can get this guy and we can get this guy. There's no parents up here for dominant for one and recessive for the other trait. No parents up there were recessive for one trait and dominant for the other trait. They were, heterozygous, yes. But when we take these guys and do the gametes, then of course you could make a 16 square Punnett square, but you guys know better to use probability to solve these. I should say this is recombinant. Now, that is the term. I don't really like it because when I think of recombination, I think of things, chromosomes breaking, crossovers occurring, recombination physically happening. But that is the term. So nobody asked me if I liked it or not. So I guess I'll have to live with it. All right. In your book, I did want to mention this. They go into crossovers involving 3 and 4 strands. Yes, those happen. We're not going to worry about them. You're not going to need that information for the basic mapping you're doing in this class. It's important and it's great to read it if you want to read it and understand how you can have triple strand or quadruple strand crossovers, but we're gonna not worry about that. Those are double crossovers, by the way, DCOs. All right, so I'm not gonna put this up there because I've talked about it. So, well, I will. In linked genes, when you have genes that are on the same chromosome, the parentals, the number of progeny that have the parental phenotypes are going to be greater than the number of recombinants. If they're not linked, it's going to be 50-50, right? 50%. If they're linked, the parentals are going to be over 50%, and the recombinants are going to be less than 50%. And so linked genes have to be close enough together not to assort independently for us to make that statement. The other things that you need to remember is that chromosomes are really, really large, especially your chromosomes. You only have 23 pairs. And so if I'm looking at a gene here, loci here, when I'm looking at a loci here, it's pretty likely that there's going to be a single crossover that always happens between them, and that they're going to be at 50% recombination. As I get closer to each other, it becomes less likely that a single crossover is going to happen every time between them. It's going to happen every time, but it may. Then I'm going to have my parentals outnumber my recombinants. Are the genes over here linked? Well, yes and no. They're linked insofar as they're on the same chromosome, but they're so far apart that they act as though they independently assort. When we build genetic maps, what we're going to do, the most accurate way to build your map is not by looking at big distances, because big distances means more and more error. It's by looking at genes that are relatively close to each other and then building a map by adding those map distances together to get the whole map of the genes. That's how we can have something that's over here at 0 and something that's at 120 over here. Yes, that just means they're on the same gene. They're 120 map units apart. So that would tell me right there, it's over 50 map units apart. So these two genes are going to assort independently. But if I come to a gene here that's at 100, then it's going to act like it's linked to that gene. So what you're observing, what genes you choose to observe makes a difference in what you'll see here. It's a long way to get around to a simple explanation. And unlinked genes, it's pretty simple. Parentals equal recombinants, as we just showed. So I'm going to do things the way I kind of learned them here. Your book has, I'm going to get to the gene pairs here in a bit, don't worry. Your book has its own way of showing this, but I like this way better. So let's start with a single crossover. I have my tetrad. I'm going to say A, B, C, D. I'm looking at 4 different loci on this guy. And of course, that means since it's a tetrad, I have this guy, or I have A, B, C, D. And we're going to make it so that I have this guy. These are sisters up here. Now I'm working on the homologs, and we're going to make the homologs all recessive. Now, if I have no crossover, you know, that can go to a gamete, 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 right? That's no crossover. But let's look at this. Actually, I should put this as, I'll do this NCO. Down here, I'm going to do a single crossover. Same idea. I like to draw it this way, and I make these drawings when I'm doing this because it just helps my brain put this together. So this is a single crossover occurring between the A and B loci. Now, there's no reason it couldn't occur here or here. I just chose that as a place where I was going to do my single crossover, where it can happen anywhere on this guy. So of course, this can go into one gamete. That's going to be parental. This can go into one gamete. That's going to be parental. But let's see what we get here. With this guy, this actually produces a dominant at the A loci, but then these two swap. So this guy is going to have all recessive. And this guy is going to start with the recessive A, and these are all going to be dominant. Okay. Because of this recombination, this swap that happened here. So what I do is I come down here and I go, oh, I just follow the cross. So this is that gamete, and I go up here, and that becomes that gamete. So that's how I do single crossovers. But as you're looking at this, the other thing this illustrates, and the reason I put more than just 2 genes up here, is that I have a crossover between A and B. But if I'm looking at B and C, I'm going to see this as a no crossover. If I'm looking at C and D, I'm going to see it as a no crossover. If I'm looking at B and D, I'm going to see it as a no crossover. Right. So it depends on what I am looking at as to far what I'm going to detect. Depends on the loci I've picked, whether I'll detect this crossover or not. If I'm working from there down, the crossover occurred, but I never detect it because I'm not looking at this A loci. Okay. So I'm going to, I took notes from your book and I put that up there. So your book uses this idea that you can use 2-point crosses to build a map. And they go through, you have all these gene pairs, and they show you in the book how they deduce that this is the correct map, except that it's not. When you use 2-point crosses to do mapping, it's fairly inaccurate compared to using 3-point crosses. So I've decided, well, let me read the shortcomings of 2-point crosses. They're difficult to determine the gene order when the genes are really close together. With a 3-point cross, it's not difficult to determine the order. And the actual distances don't always add up. So instead of teaching you all this time on a 2-point cross, I want to jump into teaching you how to do a 3-point test cross. To that end, I have given you guys a worksheet that you'll work on tomorrow and possibly as homework. So you're going to turn these in individually on a week from tomorrow, but you can work on them as a group. And what I've done in here is I've put down the process of doing 3-point test cross mapping the way I learned it. And maybe I'm just a little biased, but I think this is an easier explanation than the way your book goes about doing things. So what I have on here, instead of going through this like I was originally going to do, I'm just going to start with this guy. The first part on here is a step-by-step solve problem. I'm going to go through that with you now, maybe, yes. And then you won't need my help tomorrow. No, if you need my help tomorrow, I'll be in there, don't worry. Well, I have this up there. Let's think about if I did this as a double crossover. What if I have this situation? No, I don't want to do that. And let's put the stuff up here once. A, B, C, D. There's no line there. A, B, C, D. If I have a double crossover, this is a DCO. Again, the crossover, I could have one crossover there and there, just how I drew it. What I have here in this first guy is it's going to have the dominant A allele here. Then it's going to have the recessive B allele. And then it's going to have the dominant C and D allele because it's going to cross over back over again. Double crossovers between genes are a more rare event. So we actually can use that to our advantage in a 3-point test cross. But since they're rare events, it means we have to look at fairly large populations. Now, the populations I put in the worksheet, keep it simple, are pretty small. They'd be way too small in practice. But we can get the idea of how we do this 3-point mapping. So here I have this problem of I want to know the order of the genes and the distances between the genes. I want to be able to make a genetic map. Now, the distances between the genes, this is different than a physical map. The distances I'm making are relative distances. They're actually recombination frequencies. They're not physical distances. You can have hotspots of recombination that makes genes appear farther apart. You can have parts of the chromosome that go under very little recombination, and the genes appear closer together. So these are a genetic map is a map of relative distances. A physical map is something they couldn't do back in the day, but we can do fairly easily now. I can take a sample of DNA, send it off to whomever, come back tomorrow and have the complete sequence all figured out for me, and I can find the genes and make a physical map of exactly where they're at on the chromosome. Those are, straight distances. In genetic maps, we're looking at relative distances. So let me find a marker that you guys can see. I thought I had. I don't want to use purple because you can see it, but it will. Maybe one of my blacks up here. Do I have a better black? All right, what was I thinking about? Okay, so here's the problem. Here's your parent. You're given a parental, and we're going to do our and we're going to cross it with our triple recessive. Okay. And that's going to give us an F1, and all of the F1s are going to be male or female, doesn't matter, right? We're not talking about sex-linked chromosomes here. We're talking about autosomes. Almost sounds like AutoZone. Car parts, okay. I am off in La La Land again. Okay. So our F1s are all going to be this. Now, what we're going to do is we're not going to cross the F1s with the F1s like we would in a dihybrid cross. Instead, we're doing a test cross, a 3-point test cross, which means I want to cross it with one of these guys. Test cross is always recessive, right? So I'm going to use this. This is my test cross. Taking this guy and crossing it with something. I'm looking at 3 genes. So I'm looking at 3 genes here, all recessive here. And I know these are heterozygotic in each position because I started with pure breeding and pure breeding. Make sense? Okay. So when I do this, I get these results. I see that of the animals, flies, plants, whatever we're looking at here, that have the dominant phenotype all across, I get 360 of those guys. And then of the ones that are recessive all across, the other parental, I get 340, which sounds about right. I expect it to be around 50-50, but not exactly 50-50. Right? So these are my parentals. But then I get some that have the phenotype of dominant recessive, and of course, recessive dominant. Yeah, that's wrong. Okay, I just found a mistake in my worksheet, but it doesn't matter. We won't belabor that point. And I get 85 and 95. So let's think about this really quickly. I'm going to with a single crossover, I can either have this guy or I can have This guy, that's supposed to be on the other side of B. So I'll put this up here so it makes more A, B, C, A, B, C. These are the two options I have for a single crossover. It's a single crossover. It has to happen between one of the pair of loci, right? Keep that in mind. So this is a single crossover here. And we'll get to how we determine that in just a second. And then the next one is, let's see. Also a single crossover, and I get 40 and 30. Now, these are about evenly split, which I'd expect. There's been a crossover, but a crossover is still a fairly rare event, so it's nowhere near as high as the parental. And there's been a single crossover here as well. They're almost even, 40 and 30. And so one of these is this crossover. I don't know the order of the genes yet, though. So this is not necessarily the order of the genes. That's one of the things I have to figure out. I just have this data of what I started with. And then we have our double crossovers, which are going to be A, B, C, and A, B, C. And these guys are evenly split, 25 and 25. Now, how did I know which are single crossovers and which are double crossovers? And which are parental? Well, the first thing I know that the phenotype that's going to be the greatest phenotype is parental. That's way different than everything else. That's got to be my parental phenotype. I'm not going to worry about the single crossovers now. I know that a double crossover is a much rarer event than a single crossover. I'm having two crossovers here. So I expect it to have the lowest number of progeny, represented by the lowest number of progeny. It's kind of close in this one, but 50 is less than 70. So this has to be my DCO. That leaves these two guys as having to be my single crossovers. That makes sense? So now let's figure out the gene order. If I look at my parentals and my DCO, I'm going to forget about the single crossovers. Look at my parentals. I have all dominant, all recessive. Down here, I have dominant, recessive, dominant. The one that's different is recessive. In other words, these two guys traveled together. This one was the one that got caught in the crossover, the double crossover. And I can do the same thing here. Here I have recessive, recessive, dominant. This is where the double crossover occurred. Now, in this case, it was a sample problem, so I made the gene order A, B, C. Not all the problems are like that. But I wanted, you see how I got the gene order here, right? But I looked for which of these guys, A and C traveled together in both cases. Nobody traveled with B. B flipped down here. A and C traveled together, A and C traveled together as either dominants or recessives. The one that is different is my B. That's the one that flipped. That's the one that's in the middle. That logic makes sense. These problems are easy as can be when you practice them. But the logic's a little twisted if you just try to figure it out, the night before the exam. So it's really not that bad. You look for which one's got the most, which one's got the least, and then I look at these two and say, which one of these changed? And That one's the one that flipped. That's got to be the one in the middle. It could have been written so that C was the one that flipped, in which case I would say that it was ACB and not ABC. All right. So now I've got some good things going on here. Now I need to do some recombination frequencies. The recombination frequency here is I'm going to add these guys together, but I'm going to throw a little thing in there that we have to think about. So I'm counting single crossovers, but there was a crossover here as well. I can't not count it. Okay. So I don't have to worry about that here. So I got 360 and 340, and I'm going to put that over. I believe the total was 1,000. And right. Down here, to calculate this single crossover and this single crossover frequency, I'm going to take 85 plus 95 plus 25 plus 25 over 1,000. In other words, I got my double crossovers. I'm going to count it because a single crossover occurred there as well. I'm counting singles, but the double had a crossover. Two of them, but so I'm going to put those, I'm going to include those numbers. Now, the way your book does it is they teach you, get all the numbers, and they say, oh, by the way, there is the double crossover correction. I think you just understand there's a double crossover correction, and you put it in at the beginning. So down here, it's going to be 40 plus 30 plus 25 plus 25 over 1,000 times 100 to get you your recombination frequency. Okay. Down here, you're just looking at double crossovers. It's going to be 50 over 1,000. Right. And if you do that, 85 plus 95 plus 25 plus 25 over 1,000, you'll find out that you get 0.23 multiplied by 100, and you get 23 centimorgans. Now, you got to figure out which interval you're looking at. I know the gene order now. I know it is ABC. So since this is actually the right gene order, then I know that the crossover happened here. These are dominant, dominant, recessive, recessive. So this crossover is that first one up here. This crossover, dominant, dominant, recessive, recessive, this is the one that changes, is in this position. Right? So I can look at this and I can deduce which one of these is which crossover once I know the gene order. It's a really step-by-step kind of process here. So when I do that, and I take my 40 plus 30 plus 25 plus 25, I get 12 centimorgans. And if I make a map, I'm not going to draw it. It's a real simple map. I got 23 centimorgans followed by 12 centimorgans, A, B, and C. That was an easy map to make. And that's an accurate map because I've taken in the consideration of the double crossover because I'm using a triple test cross for mapping. That leaves coefficient of coincidence and interference. So I've got my map. I want to know what the coefficient of coincidence is. And this is pretty easy. I take, what's my expected double crossover frequency? Erase some of this. The good news is it's all written down here for you, so you don't have to catch all of my writing on the board. So the question is, what is expected DCO? Well, if I look at my map, I know there's 23 centimorgans and 12 centimorgans. Here's B here, here's A here, and here's C here. Which means that I expect a crossover to happen here 23% of the time. And I expect a crossover to happen here 12% of the time. The expectation for a double crossover would be 0.23 times 0.12. Remember the product rule? Okay, two independent events. So I'm just going to take 0.23 times 0.12, but I want to know the expected number. So my total population was 1,000. And I find out that I expect 27.6. That's my statistical expectation based on these percentages. I expect 27.6% double crossovers. I observed 50. 50 is more than 27.6. So what we're going to do is we're going to take our observed, subtract our expected, observed minus expected, and that's going to give me a COC, coefficient of confidence, written here, I don't want to write it all down, of 1.81. So how do I use this coefficient of confidence? Well, I've compared my expected double crossovers to my observed double crossovers. So now I'm going to take this COC and I'm going to say interference equals my 1.0, 100%, minus 1.81, and that gives me, what do I have here? Negative 0.82%, or 0.82. I have negative interference, and this is telling me that I actually observed more. It's negative. So if I have a lot of interference, you're going to have less because there's interference, right? So a positive interference means it interfered and I had fewer double crossovers than I expected. A negative interference means it had more. There may be a hot spot here of causing it to recombine more than expected. So based on everything being equal, my expectation was 27.6. Based on this experiment, I got 50, which might make me believe there may be a slight hot spot between these two. However, these numbers are not very big, and I would have to repeat this experiment over and over and over again and keep getting similar results before I could make that statement. That makes sense? Everybody's brain hurt now? All right. I think that is, yeah. So then the rest of this, I have practice problems. You can wait till tomorrow to do those in practice session, but I do want to show you this. The very back page is a summary of the steps. It says, identify the parentals, highest frequency, identify the double crossovers, lowest frequency, determine the gene order by comparing the parentals with the DCOs, classify the SCOs versus DCO classes. Actually, just be classify the SCOs versus DCOs. Always count the double crossovers when you're calculating map distances, and calculate the recombinant frequencies. And remember that how does that say 100 equals centimorgans? I don't know why I wrote that. I was getting sleepy last night. That's why I didn't work on your exam anymore. And compute the coefficients of coincidence and interference, and then interpret them. What you're really looking for is interference. If interference is 0, that means there was no interference and that you got 27.6 or pretty close to it versus what you expected, right? If there's positive interference, you're going to have fewer double crossovers than you expected. If there's negative interference, it's going to be more double crossovers than expected. Everybody got that? Everybody's ready to do that, ready for the exam on Friday that I have got to write. If you scheduled for the Student Success Center, man, my brain is